Hi, I'm Prasanth. I did my summer research internship in Dr. Michael Schultz's lab, Department of Radiology, University of Iowa. Dr. Michael Schultz's lab generally deals about development and use of small molecules that can be used for therapy of various types of cancer. During this summer, I did a project on small molecules that can be used for therapy of metastatic melanoma. Before getting into the topics, I would like to give a very short introduction about metastatic melanoma. Metastatic melanoma, if you see, its incidence is increasing faster than any other cancer worldwide. If you see in the past, like 75 years ago, its incidence is 1 in 1,500 persons, but now it's like 1 in 50 people. That's really a dramatic difference, I would say. It's the most common form of cancer in young adults between the age group of 20 to 30. And also, if you see, the advanced metastatic melanomas are highly lethal, as there is no durable systemic treatment for metastatic melanoma. Taking everything into consideration, our lab, Dr. Michael Schultz's lab, have decided to work to find some suitable, durable systemic treatment for metastatic melanoma. In our research, what we found out that there's a basic difference in metabolism, especially mitochondrial metabolism, between a metastatic melanoma cells and a normal cells. In a metastatic melanoma cells, which has a very high glucose metabolism, has a very high electron transport activity. In general, what happens is that as the electrons actually moves from the complex 1 to complex 3, and they actually increase the mitochondrial membrane potential. If you see, there's a mitochondrial membrane potential across the mitochondrial membrane. This is very high in a metastatic melanoma cells compared to the normal cells. So what we are trying to use is that there's a small molecule we have designed in a lab known as a triphenylphosphonium. This molecule has a very unique structure. If you see, the head region has a positive charge over the phosphorus compound. That's usually when them very good space to get translocated into the mitochondrial membrane. So this molecule, because of its positive charge and mitochondrial membrane potential, they get into the mitochondrial membrane, and it is hypothesized to inhibit the electron transport chain. So as they inhibit the electron transport chain, they are supposed to increase the superoxide species. If you see the superoxide species is increased, they can damage the cells and can bring about the death of the cells. So our hypothesis is the effective therapy of metastatic melanoma can be developed by combining the TPP-based drugs with inhibitor of hydroperoxide species metabolism. So, in order to see how effective triphenylphosphonium is in killing the metastatic melanoma cells, we have went for a first assay, which is an MTD assay. MTD assay is basically a cell viability assay that is used to see the number of viable cells after treating it with the suitable drug. So here the drug is triphenylphosphonium. Here we actually try to see the effect of 4 different variants of TPP-based drugs, that are 5, 10, and 15, and 20 triphenylphosphonium. So we conducted 2 combination of experiments, one with just a triphenylphosphonium and other with combining it with a butane sulfoxamine, which is an inhibitor of hydroperoxide species metabolism. The net result is that it increases superoxide species, and the effect will be more in killing the cancer cells. So the result is very clear that the higher orders of the triphenylphosphonium, that is a 10, 15, and 20 TPP, has a much more effect in killing the cancer cells compared to the 5 TPP, and its sensitivity is increased when we try to combine its action with butane sulfoxide. So what we understood from the MTD assay is that the triphenylphosphonium is highly effective in killing the metastatic melanoma cells, but what we don't know is what is mechanism of action. Whether it acts as an uncoupler, that is, it decreases the mitochondrial membrane potential and disturbs the mitochondria, thereby killing the cells, or it's just inhibiting the electron transport chain, and thereby it's increasing the superoxide species and killing the cells. So in order to check it out, we went for a second experiment, which is a JC1 assay, which is generally used to measure the mitochondrial membrane potential. Here we actually treated the two different sets of cells with 5 triphenylphosphonium and 10 triphenylphosphonium. The results actually showed that 10-triphenylphosphonium is actually effective in decreasing the mitochondrial membrane potential compared to the 5-triphenylphosphonium. This clearly indicates that triphenylphosphonium acts as an uncoupler, that is, it's decreasing the mitochondrial membrane potential. But what we are unclear now is that whether it's also inhibiting the electron transport chain. To check that out, we actually continued with our third experiment, which is the DHE oxidation assay, where the DHE actually oxidates the superoxide species and it fluorescence. This is a measure of fluorescence intensity. Here we did with the two variants of TPP, 5 and 10-triphenylphosphonium. And the results are such like that 10-triphenylphosphonium is much more effective in increasing the superoxide species compared to the 5-triphenylphosphonium. This clearly indicates that triphenylphosphonium is also inhibiting the electron transport chain as well as acting as an uncoupler. So what we come to a conclusion is that from our MTT assay state that triphenylphosphonium is effective in killing the metastatic melanoma cells, and its sensitivity is increased when we try to combine it with the BSO. The JC1 assay actually clearly indicates that TPP based derivative acts as an uncoupler and decreases the mitochondrial membrane potential. Our DHE oxidation assay actually clearly indicates that TPP based derivatives increases superoxide species by inhibiting the electron transport chain and thereby kills the cells. So our research concludes that TPP is a promising drug for clinical therapy of metastatic melanoma in the future. During this summer, I had an opportunity to work with other techniques and learn many techniques as well. So one such techniques I learned, the first is a chronogenic assay, which is another cell viability assay. This is actually a very time-consuming assay, so I didn't have enough data to present here. The next I did is synthesis and the purification of 5 and 20-triphenylphosphonium using HPLC. And I also had an opportunity to work with Dr. Michael Schultz to radiolabel a peptide using a gallium-68 that would be used for imaging and therapy of various types of cancer. We hope that. I would like to thank Department of Biotechnology, Khorana Program, and Indo-US Science and Technology Forum for giving us an opportunity to come to US and do a similar research internship. I would like to thank University of Iowa and University of Wisconsin for giving us an opportunity. And I would like to thank my lab members, Dr. Michael Schultz and Kelsey Coping for helping me for this thing. Thank you.